All right, so I'm going to try to do a few short little recordings of basic concepts. In this case, it's chapter 5.1 of the Pierce textbook, and it's about modifications involving one gene. I'm going to try to keep these 10 or 15 minutes, so they're not, so they're you can just quick review. All right, so if this is modifications of Mendelian genetics involving one gene. So let's first review what Mendelian genetics, classical Mendelian genetics looks like. And essentially that is with one gene, it's a monohybrid cross. So for example, if we just used a capital A to represent a dominant allele, cross it to a homozygous recessive. So there's a homozygous dominant cross to a homozygous recessive. We get a heterozygote, which is also known as a monohybrid. Cross two monohybrids together, and we get the classic 1 to 2 to 1 genotypic ratio and a 3 to 1 phenotypic ratio. The reason we get a 3 to 1 phenotypic ratio is because big A is dominant to little a. So if you get one big A, you get a particular phenotype, whatever that dominant phenotype is. And so we can abbreviate the genotype like that, where we have a big A and then either a big A or a little a. But whatever that is, that's going to show the dominant phenotype. And then this is little a, little a. So we get 3 of these and 1 of these. So that's a typical or classical Mendelian monohybrid cross. So anytime you see a 1 to 2 to 1 ratio or a 3 to 1 ratio, or some kind of modification of a 1 to 2 to 1 ratio, that means we're dealing with one gene and it's a monohybrid cross. So that's just sort of the definition. So what we're going to see with modified Mendelian monohybrid crosses is that instead of getting a 3 to 1 ratio, we're going to get some modification of a 3 to 1 ratio. So sometimes we'll see a 1 to 2 to 1 ratio where the in codominance and incomplete dominance, where the big A is not dominant to little a, and so the heterozygote, so this guy will give us its own phenotype. And then sometimes we'll see situations like a 2 to 1 ratio where one of these categories doesn't show up at all. So we'll take a look at that. So over here on the left are some of what's covered in this little part of this chapter. And so when we're talking about modified genetics with one gene, we're going to talk about incomplete dominance and codominance. We'll look at some problems that involve those concepts. We'll briefly touch on penetrance and expressivity, just sort of a definition of those things. Lethal alleles is an example that we'll look at, and then we'll also look at these other two concepts. And hopefully we can get all that done in less than 15 minutes. All right, so let's start with incomplete dominance. So incomplete dominance is very similar to codominance. And so we'll just look at an example of this. The classic example is in snapdragons, which is a plant with flowers. And if you cross a red snapdragon to a white snapdragon, you get pink snapdragons. So that tells us that red is not dominant to white, because if red was dominant to white, then it would be all red. But we get all pink. But we don't really know what's going on yet. To figure out exactly what's going on, we have to do the next cross. So this is, in standard Mendelian genetics, we have a parental cross, an F1 cross, and then we look at the F2. So if we do a self cross of two pinks together, if it was a normal Mendelian genetics, we would expect 3 to 1. But we don't get 3 to 1, we get 1 to 2 to 1. And these are red, and these are pink, and these are white. And so if it's a 1 to 2 to 1 ratio, that means it's a monohybrid cross. So that means that these guys, if we used classic symbols, these guys would look like that, and then maybe red would look like this, and white would look like this. And down here, red would look like this, pink would look like this, and white would look like this. Now that's okay, but there's a problem with that symbolism, because by definition, a capital A is, or a capital letter to represent an allele is considered dominant. But in this case, the big A is not dominant. So we have to give it another kind of designation. So instead of using capital letters, we use the same letter to represent the gene. So instead of capital letters, we'll go with red and white. And instead of indicating that one thing's dominant over the other, we can sort of keep it neutral, and we'll give. So the locus or the gene will be called the C locus, or the C gene for color in this case. And if it's red, we'll give it a superscript R. And if it's white, we'll give it a superscript W. So this is just kind of standard genetic notation, where we often have the gene name or the gene symbol, and then the allele symbol is the superscript. So the gene, so remember, on a chromosome, you can have a locus or a gene, and you can have different alleles at that locus. And so maybe this is the C locus or the C gene. And if we are homozygous for the red allele, we would look like that. So that's a superscript. And if we were heterozygous, we could be heterozygous. We'd have the C gene and the R allele or the W allele. So it's just a different way to represent different versions of that gene. Remember, an allele is a different version, or things that are alleles are different versions of the same gene. And that means they occupy the same locus, the same position on the chromosome. We cross these together and we get a heterozygote. Cross two heterozygotes together and we get a 1 to 2 to 1 ratio. And if you feel like doing a Punnett square to prove this, feel free. And these guys are red. These guys are pink. These guys are white. So anytime you see a 1 to 2 to 1 ratio, that indicates that one gene is involved. That's critical here. So later we'll look at 9 to 3 to 3 to 1, and that's always a 2 gene situation. But 1 to 2 to 1 or 3 to 1 or some modification of that is always one gene. That's sort of a definition of one gene. So I'm going to move this up here. All right, so now let's look at, so that was incomplete dominance. Now let's look at codominance. Let's go back just for a second. So the other thing that's key about incomplete dominance is that this pink is intermediate between red and white. So somehow the phenotype is intermediate between the two original phenotypes. Codominance is very similar, just one little difference. So codominance, we're still going to get a 1 to 2 to 1 ratio. So the classic example for codominance is blood type. So if we take type A, somebody with type A blood, and we'll do a little bit more about this in a minute, but sort of classic example is this. And they're going to, in this case, they're both homozygous for an allele for each thing, and I'll show you the symbols in a minute. Then here you can get type AB blood. Now this is usually in humans, so we don't really do a test cross or like a self cross and that kind of thing, but just sort of conceptually, if you cross these two together, we'll get 1 to 2 to 1 type A blood, type AB blood, and type B blood. All right, so what's going on with that? So in blood type, there are a few alleles, 2 of those alleles. So just like we had the color, like the C for the color locus before, we're going to use a capital I for this. Don't ask me why. It's just complicated. I don't know. It seems like a dumb letter to use, but that's what they use. And if you have this, you have type A blood. So the phenotype is the type of blood you have. So type A. And if you have this, you have type B blood. We'll add another wrinkle to this in a minute, but right now we're just talking about these alleles. And then if you have a heterozygote, you have AB blood. And that's it. And we can tell, so that means that this guy has this genotype. This guy has this genotype. And here we're just talking about codominance. And then these guys have this genotype. And these guys have this genotype. All right, now, so how is that different than incomplete dominance? Well, it comes down to what's happening at the molecular level. For incomplete dominance, we won't worry too much about the molecular level of what's happening there. But here, we can think about what's happening at the molecular level, or at least the cellular level. So here's a cell, and this is in a blood cell, a red blood cell. And you've all heard of blood type. You have whatever types you have. We haven't talked about type O, but we'll talk about that in a minute. And what blood types are is that there are specific carbohydrates that are on the surface of the cell, and they're called antigens. And so let's say that type A antigen, we're going to give that a red dot, and type B will give a blue dot. And so what that is, it's really a different carbohydrate that is on the cell. And so let's make 3 different types of cells. And we'll start with type A. We'll have type B and type AB. So type A has a particular type of carbohydrate stuck on the surface. So that's type A blood, because it only has the A allele. The A allele makes the red carbohydrate in this case, and it only has that. Type B makes a blue carbohydrate that gets stuck on the cell surface. And type AB has both. And so these carbohydrates, that's supposed to be red. These carbohydrates are added to here by an enzyme, which is a protein. And the protein is made by a gene, and different versions of that gene are the A allele and the B allele. If you have the A allele, you get the red carbohydrate. If you have the blue allele or the B allele, you get the blue carbohydrate. And if you have both, you get both. So this isn't intermediate. So what's intermediate between red and blue might be purple, right? So instead of getting purple, we get red and blue sort of spotted. So we get both things showing. So neither thing is dominant to the other. Dominance means if you are heterozygous, so remember, dominance means that if you have this or this, you get the same result. But here, we're not getting that. If you get homozygous, you get one result. If you get heterozygous, you get a different result. And so neither A or B is dominant. If A was dominant, then if you had an AB genotype, you would have an A phenotype. If B was dominant, you'd have a B phenotype. So that's what codominance means. But the key here is to look at the pattern. Because we see a 1 to 2 to 1 ratio, anytime you see a 1 to 2 to 1 ratio, that's a monohybrid cross or a modification of it. So let's go on with blood type a little bit more. And so we'll add another page, and then I'll move it up in a minute. All right, so there's more than 2 possible alleles for the blood type. So this introduces a concept called multiple alleles. So there are 4 blood types. So we've got we just talked about type A, type B, and type AB. We also know that there's type O. So type O, there's another allele called the O allele. And if you're homozygous for the O allele, you get type O blood. So this is the phenotype. This is your blood type. The genotype would be I superscript O, I superscript O. A and B are codominant, but O is recessive to both A and B. So that means if you have type A blood, you can have two A alleles, like we talked about before, or you can have an A allele and an O allele. B, you can have two B alleles, or a B allele and an O allele. AB, there's only one way to get AB, and that's if you have both alleles. So because, in this case, when we're heterozygous for A and O, we get type A. When we're heterozygous for B and O, we get type B. That means that O is recessive to A and O is recessive to B. And so the only way to get type O blood is if you get two O alleles. So because now we know that that allele is recessive to the other ones, we can actually use another symbol called little I. So lowercase I. Because before we talked about a capital letter means dominant and a lowercase letter means recessive. The O allele is recessive to everything else, so we can give it a little I. So now we can describe this as what we call an allelic series, which describes the dominance relationship between different alleles of the same locus. So here we know that the A allele is codominant with the B allele, so we say it's equal to the B allele. But both of these are dominant to the O allele, so we can say greater than, it's a greater than sign. So by knowing this dominance relationship between these different alleles, we can predict the outcomes of crosses. Or we can, like for example, if we have somebody with type A and we cross it with somebody with type B, and we get out of that, we get some A, we get some B, we get some AB, and we get some O. It's like, well, there's a lot of stuff going on there. But we know in order to get O, we need to have two of these. So if we need to get two of those, as this is offspring from this cross, we wouldn't draw it as a cross like that. We draw it as a pedigree, because this is blood types in humans. But if we have two little i's, that means we know the genotype here. We know that this guy had to have a type A allele to have type A blood. And we know that it could be homozygous for A, or it could be heterozygous for A and O. And this guy we know has to have a B allele because it is type B blood. It could be BB, but if it was BB, we would never get these O's down here in the offspring. So that means it also has to have an O allele. So now it's possible to get, so this type A here would have to be this. Type B would have to be this. Type AB would have to be this. And type O would have to be this. And so you can, by knowing this allelic relationship, you can predict the different types of outcomes from this kind of a cross. So there are some problems that talk about this, so I would look at those problems, and we can look at some of those in a bit. But I don't want to spend too much time because this is just supposed to be quick. So that's what we've done so far. Is we've talked about incomplete dominance and codominance, and now we've added this multiple allele concept. Now, another concept I want to talk about is lethal alleles, and so that's another modified monohybrid cross. So lethal alleles, and the classic example for this is in Drosophila. is if you take two curly winged flies, you always get a 2-to-1 ratio of curly to straight. And straight we're going to call wild type. So wild type always means sort of the normal type of wing or the normal phenotype in whatever organism. So for this gene, we have a straight wing or we have a curly wing. Kind of looks like that. And so what can we learn from this? Now, we didn't cross curly to wild type here because it turns out that if you do cross curly to wild type, you always get 1/2 curly and 1/2 wild type. So how would we figure out this sort of pattern of inheritance? These are really the only, if you cross wild type to wild type, you always get all wild type. So what's going on here? And you never get a case where you cross curly to wild type and get all curly or anything like that. So how do we interpret this? Well, first we see this 2-to-1 ratio, and we have to think, well, that is sort of like a 1 to 2-to-1 ratio. It's just missing the 1. And so that's what's going on. So if it's a 1 to 2-to-1 ratio, and keep in mind this chapter section is about modified Mendelian ratios, and we're looking at modified monohybrid crosses now, that means that these two curly guys are heterozygous. How do we know that? Because this is a 1 to 2-to-1 ratio, even though we can't see the 1 yet, which I'll explain in a minute. If it's a 1 to 2-to-1 ratio, that means it's a monohybrid cross. So that means we'll just use C for curly. So that means that big C and little c cross to big C and little c. It's a monohybrid cross, so it has to be two heterozygotes crossed together. That's what a monohybrid cross is. So we say C equals curly and little c equals wild type. How do we know that? Because we know it's a monohybrid cross, which means we know these parents here. We know they are heterozygous because it's a monohybrid cross, and that's what a monohybrid cross means. And if they're heterozygous and they show a curly phenotype, that means that curly is dominant. We also know I didn't leave enough room for a 1 up there, so I'll put it down here. So we know, well, let's do it over here. So let's say we have a monohybrid cross. We get a 1 to 2-to-1. But here we're getting a 2-to-1. So we're not seeing something. One of these 1s is gone. And so it turns out that this one dies. So in this case, I just used A's to show a normal monohybrid cross. But when you do a normal monohybrid cross, the 2 is always the one that's heterozygous. So up here, we know that this 2 is heterozygous. And we also know, because it's curly, it has to have that dominant allele. So what's going on here is this guy is big C, little c. This guy is little c, and then the big C would be the other 1. So it would be 1 to 2-to-1. But these guys die. So we don't see these. So if we do this cross and we just count the flies that we see, we'll get 2/3. So we would see this as 2/3 curly and 1/3 wild type. And that's a classic example of a lethal allele. If you see a 2-to-1 ratio, it's a modified 1 to 2-to-1 ratio, and it's called a lethal allele. So that was the other example there. So the other couple of things that we need to talk about, let's look up here and see what our list was. So we talked about incomplete dominance, codominance. Complete dominance just means normal Mendelian, one thing dominant to the other. We haven't talked about penetrance and expressivity yet. We talked about lethal alleles. And we talked a little bit about multiple alleles. We could maybe do another example of that. And then we haven't talked about a compound heterozygote, but that's pretty simple. We'll talk about that in a minute. So we'll do Now we're going to do penetrance, variable expressivity, and what a compound heterozygote is. So penetrance. Simple definition. We see this a lot in pedigrees. And basically what that means is that if somebody, let's say there's a disease, so we have a pedigree, and we have two individuals that are heterozygous, and they have some heterozygous, some homozygous, and they have some homozygous recessive. Then let's say, so I just put those genotypes in there. So let me give them their little symbols here. And it doesn't matter if they're male or female, we're just putting in whatever. And this person is affected with the disease, and this other person is not. That means that there's not, so if it's 100%, so that means it's less than 100% penetrance. Now, we don't know that it's 50% penetrance at this point because we've only looked at two individuals. But a given disease genotype can have less than 100% penetrance. That just means that everybody who gets that genotype doesn't necessarily get the disease. So you either get the disease or you don't. You could have two people that are both little a, and they don't get the disease. So the way problems look like for this is you say there's a recessive disease that has 30% penetrance. So what that means is that 30% of the people that get little a don't get the disease. Now, the reasons for that are basically sometimes there's environmental factors that are involved or other genes that interact. There's just the sort of genetic background of the person plays a role. But the way that, let's say you know that. Let's say you know that there's a disease with 30% penetrance and it's recessive, and you get something like this where somebody has a disease, the other kids don't, and you want to know what the probability is that this next person is going to get the disease. Well, because one person has a disease already and neither of the parents have the disease, we can say that, well, I guess this isn't, because of the penetrance thing, it's not 100% true that we know this yet, but so let's pretend we know that the two parents are heterozygous. I guess they have to be heterozygous if this first kid is getting the disease. And these other two don't have the disease, so we don't know their genotypes necessarily. But then what's the probability of this fourth person getting the disease? Well, if we know it's recessive, we know there's a 1/4 probability. We'll say 0.25. 0.25% or 0.25 probability to get that genotype. But of those people, only 30% will get the disease. So that's the probability of getting the genotype. The probability of getting the disease is 30%. So the way we would figure this out is 0.25, the probability of getting the genotype, multiplied by, like, if you get the genotype, the probability of getting the disease is 0.30, because 0.30 is the same as 30%. And so we would just have to figure out what 0.25 times 30 equals. Now, I don't have my calculator handy, but that's 1/4 of 30. 1/2 of 30 is 15. 1/2 of 15 is 7.5. So we would say there's a 7.5% or 0.075 probability that you would get that disease. So that was just a little mental math. You would be welcome to use a calculator. So it's just adding in that penetrance stuff. The next concept was incomplete or variable expressivity. And that just means, so variable expressivity means that the phenotype expressivity so let's say you have two individuals that that both have this disease, but the severity varies. So this is severe, and this person has a mild case of the disease. And that's the case with most diseases. Like even like if it's cystic fibrosis, for example, let's say you have cystic fibrosis running in a family, and we know that both of these people have the disease, but this person has very much worse symptoms, and this person has much more mild symptoms. That's basically all it means, is that the phenotype, whatever it is, even if it's like in fruit flies, you could have some kind of wing phenotype where you have a long wing, and then you have a mutant where you have a short wing. But sometimes, you can get kind of all of these. So this would be wild type, and each of these would be some kind of mutant, but they'd all have the same genotype, but they'd have different variations or different levels of expression of that mutant phenotype. And so that's all variable expressivity means. And then the one last concept that it talked about was heteroallelic combinations, or maybe they called them compound heterozygotes. And what that means is, let's say you have somebody with two people are carrying a mutant allele for cystic fibrosis, and they have kids. So both of these parents are heterozygous for a mutation in the gene. So let's pretend this is the gene, the cystic fibrosis gene. But there's more than one possible. So in the population, there are lots of different cystic fibrosis alleles. That just means, and so if this is the gene, let's say that's a normal gene, call that wild type, which gets a plus. Then here's a mutant CF allele 1, where they have a mutation over here in the gene. And then over here, there's some other allele that has a mutation. Both of those mutations disrupt the gene, but they're in different places in the gene. So even though we haven't covered it yet, you know that genes are made out of DNA, and you can have mutations in different parts of the gene. And so we would see that. So maybe this person has allele CF1, and this person has allele CF2. And then if they have a kid who shows a disease, they are mutant for both of those genes, but that's called a compound heterozygote, as opposed to being homozygous for the same exact mutation. So this is heteroallelic combination because they have two different alleles of the same gene, and so they're heterozygous for those two different things. So I'm going to leave it at that. There are longer videos on the course web that go through problems, and a lot of those problems are solved. So I'll let you work through those because I want these to be pretty short videos for now. So I'm going to leave it at that, and then I'll do the next part of chapter 5 in a different video.